All right, let's talk about our genes. We all have this idea of genetics being a simple blueprint for life, right? But the reality is, well, it's way more complex and honestly, a lot more fascinating. In fact, our whole understanding of how genes work is completely changing as we speak.
So, to get our heads around this, let's start with something fun. Something from a world a lot of us grew up with. Harry Potter. In that universe, being a wizard is in your blood. So it makes you wonder, is there a gene for magic? It's actually a perfect way to look at how we think genetics works.
Okay, so if we just slap a simple model on it, let's say the magic gene is dominant, we immediately hit some major snags. I mean, how do two magical parents have a non-magical kid, a squib? An even bigger head scratcher. How did two non-magical parents, muggles, have a super powerful wizard? These little paradoxes show us something really important. Genetics is almost never a simple on or off switch.
To really get a handle on all this complexity, we've got to start with the basics. So let's rewind a bit with our first section, the simple code. This is all about that classic model we probably all learned back in school.
You remember this stuff, right? We get one version of a gene, it's called an allele, from each parent. So if you get an allele for brown hair and an allele for red hair, you're probably going to have brown hair. Why? Because that brown hair allele is dominant. It just kind of talks over the instructions from the recessive red one. Simple enough. But as we just saw with our wizarding example, this simple model just can't explain everything.
Yeah, that simple model, it's really just the first chapter of a much bigger story. So in our next section, Beyond the Blueprint, we're going to tackle this really common myth of there being a single gene for any given thing.
You hear it all the time, right? A gene for intelligence or a gene for being a great athlete. The thing is, that idea is a massive oversimplification. In the real world, having one single gene responsible for one single trait is incredibly, incredibly rare. Most of the things that make you you are the result of tons of genes, all working together in these complex interconnected networks. So, if our genes aren't a blueprint, then what are they? Well, the biologist Richard Dawkins came up with a much, much better analogy. Our genes are like a recipe. See, a blueprint tells you exactly what the final house will look like, down to the last detail. But a recipe gives you ingredients and a set of instructions, and the final cake just sort of emerges from that process. The cake's structure isn't spelled out line by line in the recipe.
And this is why that recipe analogy is just so perfect. There is this long, complicated journey from a single gene to an actual physical trait. A gene doesn't just, make blue eyes. No, it kicks off this whole chain reaction, building proteins which form tiny structures inside cells, which make up the cells, which form tissues, which all come together to make the final organism. It's a process.
Now, are there exceptions to this rule? Oh, absolutely. And they're pretty cool. For instance, get this. There's a short sleep gene. Somewhere between 1% and 3% of people have it, and it lets them feel totally rested on just 4 to 6 hours of sleep a night. But while these single gene effects are fascinating, they really are the exceptions that prove the rule. The big story is complexity.
And all this complexity brings us to a huge, huge piece of the puzzle. It's the part that explains how this same recipe can be interpreted in so many different ways. Section 3, the ghost in our genes. We're going to talk about a hidden layer of control called epigenetics.
So, what on earth is epigenetics? Well, that prefix epi just means on top of. So, it's the study of changes that affect how your genes get used, but, and this is the key part, without changing the DNA code itself. Think of it like this. Your DNA is the computer hardware, but the epigenome is the software that tells the hardware which programs to run and when.
So how does the software actually work? I mean, on a physical level. To get it, you have to picture what our DNA really looks like inside our cells. It's not just floating around like a long, tangled piece of paper.
Instead, it's neatly wound around these little protein spools called histones. Think of it like thread on a spool. Now, these epigenetic tags are basically little chemical marks that attach to the DNA or the histones. They can tighten the coil, which hides a gene and turns it off, or they can loosen it, which exposes the gene and turns it on. And get this is the incredible part. Your lifestyle, what you eat, the environment you live in, all of those things can influence these tags, constantly changing how your genetic recipe gets read.
Okay, so once we started to really understand this, that genes are a recipe, and that there's this whole other layer of control on top, it led to an absolutely mind-blowing leap in technology. Which brings us to Section 4, rewriting the recipe. Let's talk about the power of modern genetic engineering.
The technology that basically blew the doors wide open is called CRISPR-Cas9. The easiest way to think about it is like a pair of molecular scissors that has a GPS built in. It lets scientists find a very specific spot in our massive genome and then make a precise cut or an edit. It's incredible. For the first time in history, we can actually go in and rewrite the letters in the recipe itself.
But with this incredible power comes a really critical distinction we have to make. On one hand, you have somatic therapy. That's when you modify the cells of one person to treat their disease. And that's pretty widely accepted because those changes die with the person. But on the other hand, you have germline editing. That's when you alter the DNA of an embryo. And that is way more controversial because those changes are permanent. They're heritable. They'll be passed down for all future generations.
And that massive debate is exactly where we're going next. It brings us to our final section, which is all about what comes next. Section 5, our genetic future, and the really profound ethical choices that we are all facing right now.
So let's just break down the big conversations that are happening. When we talk about modifying humans, critics warn about creating a genetic elite, a world of haves and have-nots, and a very slippery slope to eugenics. Then there are gene drives. These could wipe out diseases like malaria, which is amazing, but they could also cause irreversible damage to entire ecosystems. And then there's the whole issue of corporate control over GMOs, which raises these deep questions about who gets to own the building blocks of life itself. Now, our role here isn't to take sides, but just to show you how high the stakes really are.
So, here we are. We've gone from just learning to read the genetic code to actually being able to rewrite it. The recipe for life is, in a very real way, now in our hands. And that leaves us with a profound question, one that every single one of us has to consider. What kind of future are we going to choose to create?